Hi everyone, this is Mr. Sinti, and I want to talk to you now about how DNA replicates in a process called DNA synthesis, or making more of DNA. The interesting thing is when the structure of DNA was determined in the early 1950s, in 1953, I believe it was, by Jim Watson and Francis Crick, right after they published that famous paper on the double helix, they put out another paper hypothesizing a way in which DNA would be replicated, because it's kind of interesting. The structure of the DNA kind of lends itself to an explanation of how the molecule could replicate and pass on to cells when they divide. And as it turns out, in some ways it's rather easy, and in some ways there's some complexity. But the original molecule of DNA, as you can see here, the double-stranded molecule that's held together by hydrogen bonds, is opened up. And when the molecule opens up like this and those hydrogen bonds are broken, the light blue represents new nucleotides. So in other words, these down here, I'm sorry, let me go back. These new nucleotides here, shown in light blue, can attach. And so at the end of replication, what you get is one complete molecule and a second complete molecule. So it copies itself. And do you notice how the dark blue represents the original parental strand? Or sometimes it's referred to as a template. So the parental strand opens up and the new nucleotides come in. Where do those new nucleotides come from? They come from the cell. How did the cell get them? Well, we consume them when we eat food. When we're eating strawberries, for example, strawberries have DNA, and so we can digest them, and then the nucleotides come into the cell, and next thing you know, strawberry nucleotides are going to be part of our DNA. So what you can see is there's two copies. One copy is old and new, and the other copy is old and new, if you will. Sometimes this is referred to as semi, since it's half and half, semi-conservative. It's a good way to replicate to make sure that there's not a lot of errors. You're pairing up the sort of the new person with the old person, or a master and an apprentice. And so that's kind of an interesting point. So you can see in more detail here. Here the nitrogen bases are actually lettered, A with T, C with G. You can see that. Here's the parental strand. They open up. And then the new nucleotides come in and they simply attach with hydrogen bonds. G with C, A with T, T with A. So you can see how there's two molecules? If you look closely at the two molecules, they're exactly the same as what you would expect. If DNA is going to replicate itself, you would have two exact copies. And so this is kind of neat. If I were to draw a cell like this, the whole point is when you replicate DNA is that one of the new DNAs goes to one side of the cell and the other one stays over on this side. Because the point is, if the cell is about to divide in two, let's do it like this, then one cell will have a complete DNA and the other one will have a complete DNA. So it's rather clever. It might be obvious, but I'll state it anyway. DNA has to be replicated before the cell divides. And then you might recall from biology, there's a whole big discussion about how DNA is really huge. This is not the whole molecule. It's rather long and it's made up of many strands, and so the cell takes care to take all that DNA and wrap it up, coil it over and over and over, sort of like tissue around paper, around cardboard to make up tissue paper or toilet paper. Those big condensed rolls of DNA are called chromosomes. So the chromosomes move right before the cell divides, and that's called mitosis, if you recall that. So here's another cartoon drawing of this, a little bit more realistic even, because as you can see, here's the new helix forming of one completed molecule, and here's the other one. And these are the free-floating nucleotides. So what's interesting is, like anything in the cell, chemical reactions may occur spontaneously, but they're very slow. And so you might predict right now that where we're going in this conversation is we want to talk about what are the enzymes that are important in speeding up the replication of DNA, and there's some really important ones, as you might imagine. And so one last thing before we get into that is here's a close-up of an adenine trinucleotide. So that's one of the nucleotides. The enzyme that is going to catalyze the elongation of the new DNA, in other words, add the new nucleotides, back to the previous slide here. The enzyme, if I were to superimpose an enzyme on top of this and do this, like what's the enzyme that's going to sit on the DNA and add these new nucleotides, in other words, replicate DNA? The answer to that is an enzyme appropriately called DNA polymerase. DNA polymerase is appropriately called because it's an enzyme, you can notice by the ACE, a protein that makes DNA polymers. So it's called DNA polymerase. It catalyzes replication. One might consider it one of the most important enzymes of all in biology. And so what it does is it needs, this is just one of them, this is DATP, but there's DCTP, DTTP, and DGTP. There's 4 nitrogenous bases involved here, and so the polymerase grabs them like this. Cartoon's kind of cute. The enzyme comes along, and as you can see here, it looks like it's adding either an A or a G. I can tell that because it's large in structure. And so it's going to be bonding right here with something like, if this is A, let me just see if I can draw that. If this is A right here, the enzyme itself doesn't need to form the bond here. That's just going to be hydrogen bonds. That's just naturally going to happen. What the enzyme does need to do is bring in the triphosphate. 2 of those phosphates leave, and then the third one connects there. And so it adds new nucleotides to the previous nitrogenous base, which is over here. And this is a detail, but I want to emphasize it if you don't mind. It adds it to the hydroxyl group. And so this is the 3'. So what we're saying is the enzyme is able to add DNA nucleotides in a growing 3' direction. So the next one's going to go off on this side as well. That might be more clear in the next few slides. DNA polymerase doesn't work alone. How the molecule actually uncoils or unzips, how the helix unravels itself, is that a molecule, again appropriately called DNA helicase, shown here in what look like purple grapes, attach onto the molecule, and by attaching onto the double helix, it simply provides an ability for the molecule to open up. And once the molecule opens up into what's called a replication fork, like a fork in the road there, the hydrogen bonds break. It's rather easy. It's kind of interesting. People sometimes wonder about how can this molecule that's so important, a ladder, be held together by these weak hydrogen bonds. And I think the answer to that might be in the fact that you want the molecule to open up because you want to be able to replicate it, and also you want it to be able to transcribe it into RNA. So it's like a book, needs to be able to be opened in order for it to be read. And so again, I want to emphasize this anti-parallel structure again, because one side is the 3', the other one's 5, the other up here is the 3' and the 5. So what this means is that when the molecule opens up, say this is the parental template, when it opens up, the replication is going to be going in one direction and the other one's going to be going the other direction. And so DNA polymerase can only add at this 3' end because it's the hydroxyl group that is making the connection to the next phosphate. So it only grows in a 3' direction. Let's take a look at that in detail. So here's the template. Now, what have we looked at so far? I've mentioned 2 enzymes. I mentioned this one, helicase. What does it do again? It helps to open the helix. So here's the helix opening up. I also mentioned the enzyme DNA polymerase, which is not even being shown right here. Let me add it, DNA polymerase. What does it do? It adds new nucleotides. But what's interesting is, this is a bit of a detail, but I'll mention it. There's an enzyme called topoisomerase, which upstream of where the molecule opens up, it attaches to the molecule and prevents kinking occurring in the molecule when this unwinds. And then there are these proteins called single-stranded binding proteins, as you can see here, that attach, which try to prevent the molecule from re-annealing or getting the hydrogen bonds. Once it opens up, it's going to have a tendency to want to close back up again, and these sort of prevent that temporarily. Now, what's going on over here? There's an enzyme called primase. It's sometimes called RNA primase. And what's interesting about this is that one of the limitations with DNA polymerase is that, I mentioned this before, it can only add new nucleotides in a 3' direction, and you need to have a nucleotide there already before it could add it. So in other words, if this is DNA nucleotides floating in the nucleus, this is like A, T, C, G, A. Let's do that. What has to happen is this RNA primase puts down a little bit of RNA in the molecule. It attaches this first, sort of like a primer. Like if you wanted to paint a room blue, you would want to paint it white first and then paint it blue on top of the white. This RNA has to be put down, and then the DNA is added by DNA polymerase, like this. And so it would be growing in a 3' direction. And likewise over here, if you put down some RNA, and let me make the RNA red, put down the RNA like this. This is the 3'. Then the DNA can be added like this. And so then what happens is you've got DNA with a little bit of RNA, and then another RNA polymerase comes by and removes the RNA. I know that sounds tricky and perhaps apparently unnecessary, but it's going to be important because this whole idea of putting a primer down is going to be crucial in some of our experiments that we're going to be doing in class, and so I want to bring that to your attention. And so DNA can't initiate the synthesis of the polynucleotide by itself. It has to have a primer there first, and the primer's in the cell. The primer is RNA, and it's about 10 nucleotides long. It's that little red piece. And again, the enzyme is called primase that does this. And so let me go up here and show you a little bit. Let me come up here and see if I can, whoops. Let me see if I can grab that. Come on over here and grab the, take that off. The final thing I want to say is, let me see if I can illustrate this. So if DNA opens up like this, and it opens up like this, make a connection here like that. And then here are the exposed nucleotides of DNA, like this. And I'm just going to say arbitrarily, if this is 3', this would be 5, and this would be 3 here, and this would be 5 here. So what's going to happen, just to review this, is RNA is going to be put down, so a little piece of primer, and a little piece of primer is going to be put down over here. This is about 10 long. And so what's interesting is, if this is the primer going in this direction, this is 3, whoops, I'm sorry, that is a mistake, right there. Let's go, let's make the new ones blue as well. So if that is 5' to 3', then this would be, on this side, 5' to 3'. And so what would happen is the DNA would be growing in this direction, like that. And then over here, this would be 3' and 5, whoops, 5'. And so the DNA would be growing in this direction. So what's interesting is that they grow in opposite directions of one another. I just wanted to point that out. So I hope you enjoyed our conversation about DNA replication, otherwise known as DNA synthesis. Thanks for watching.